Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear students, I am a friend, commenter of Botany, and we have already started with our lecture, that is sexual reproduction in flowering plant, in this lecture series. Dear student, we have already studied in this chapter about the male reproductive organ, that is stamen, its structure, and how the pollen grains are produced. Within pollen grain, how male gametes are produced. Apart from that, we have also studied with the female reproductive organ, that is a carpel or a gynoecium, and in that also we have studied how the ovule structure is there, how megasporogenesis lead to formation of megaspore, and how egg cell is produced within embryo cell. Then we have also studied pollination and different types of pollination. And then we have particularly already discussed some part like fertilization. In that also we have studied syngamy and triple fusion. And after fertilization part, we are at a point where we are going to study something new things in this chapter. And in this lecture, we will be dealing with the artificial hybridization, then topic like endosperm formation, types of endosperm, embryo development. These topics we are going to study in this chapter. So students, if we talk about the particular thing, then we come to know that we are going to talk about first artificial hybridization. In artificial hybridization, the term itself suggests artificial means man-made and hybridization means we are going to cross between the two different crops. Now if here artificial hybridization is going to happen, it is one of the major approach. It is one of the major approach, dear student, for a crop improvement program. And in this approach, we will talk that how this process occur and that can be carried out. And if we talk specially of this artificial hybridization, it is one of the important technique which makes us sure that we will be crossing only the desired pollen grain with the desired stamen. So the pollination will be occurred on the basis of our will. So one more thing we can hear, that stigma is also protected from the contamination from the unwanted pollen. Dear students, here only desired pollen is going to reach the stigma. Any other pollen cannot reach. Okay. So based on this, if we talk how this artificial hybridization occur, that we have to discuss. Now see, dear students, if we talk about a flower, in a flower you know that there is a presence of stigma, ovary and style, that is carpel. Then we have a particular stamen here. We have a carpel like this. Okay. This particular flower also have a sentence. Now this is, if we talk, is a bisexual flower. And in bisexual flower, both stamen and carpel are present. But if I want to carry out the pollination to the stigma of a desired plant, the need of first thing is to remove the particular stamen or anther we can say. So in this artificial hybridization, first step which is going to be taken, that is emasculation. Now emasculation is nothing but it is removal of anther from the bisexual flower. So if we remove the anther, what will happen? We will have a particular flower with the desired carpel only. Okay. Now what I will do, or we will do, we will bag it. We will cover it one bag. Why there is a need to cover with the bag of a particular butter paper? Because if we do not cover a bag, the unwanted pollen grain can come and can bind with this particular bud. Unwanted pollen grain can come and can attach to this stigma and contamination can happen. So to prevent that, we need to carry out add a bag, and this phenomena is known as bagging. Now after bagging, next what we'll do, we will allow this in the bag, we will allow this stigma to get matured. After this bagging, and when a stigma get matured or carpel get matured, we remove the bag, and this removing of a bag is known as debagging. What it is known as? Debagging. Now after debagging, what will happen? The stigma are matured, so we can dust a particular desired pollen grain over this. So we particularly carry out dusting, and this dusting is done of pollen grains, and that pollen grain are desirable. When the desired pollen grain are dusted over this, we need to carry out a pollen crystal interaction, and we need to allow them to carry out what post-pollination event, so that we need to bag it again and allow that process. So rebagging is done. After rebagging, we will allow them to fertilization, and proper fertilization, proper cross hybridization will happen between the desired pollen and the stigma. After rebagging, we will add a one tag over that, and this tagging will indicate that a particular cross hybridization has happened. So in a bisexual flower, to carry out artificial hybridization, first we need to perform a particular process, that is emasculation. Then the bagging will be performed, then debagging will occur, then dusting will be carried out, then debagging will occur, and then tagging will occur. You will get a question from this of a correct sequence, identification of correct sequence. But dear students, we have to mark one of the important point here, is that if there is a unisexual flower, emasculation is not required. So no need of emasculation in unisexual female flower. Okay, so this way the whole process will occur, and I hope you understood this point as well. So this is the correct sequence how artificial hybridization is going to happen. So here you can see this is the cutting of specific anthers, that is emasculation. So we can understand this particular terms with the technical language, and we can know that this artificial hybridization is achieved by the emasculation bagging. If the female parent bear a bisexual flower, removal of anther from the floral bud, we remember this removal of flower for this particular anther occur from the floral bud. It is done in the floral bud. Okay, before the anther days, before the removal of before the anther release their pollen grain, we have to cut them by the pair of forceps. That is necessary, and that is known as emasculation. So what you have to understand, emasculation is done before the release of pollen grain by the anther. We have to remove the anther from the bisexual flower. Then these emasculated flowers have to be covered with the bag of suitable size, generally made up of butter paper, prevent a contamination of its stigma with the unwanted pollen, as we have studied. Know this. Then when the stigma of a bagged flower attain receptivity, they get mature. Pollen grain collected from the anthers of male parent, that is what we call a desired pollen, are dusted on the stigma, that is dusting, and the flowers are rebagged. After rebagging, this particular fertilization will happen. Later on, embryos will develop and fruits will allow to be developed. If the female parent produces a unisexual flower, then we have to be cautious that there is no need of emasculation. Okay, so this is the thing. Now, so let's talk about one question. Removal of anther from the bisexual flower is called as, you know very well, I must say that you are sure about this, and that is emasculation, when it occurs in the bisexual flower. Next to that, another question is, emasculation prevents desired self-pollination? No, it cannot prevent, because emasculation is particularly need a particularly desired cross-pollinated plants. So yes, but why we emasculate? We have to add a desired particular pollen grain, and we have to perform cross-pollination. But it prevent what? Undesired, means self-pollination. Undesired self-pollination it can prevent. Apart from that, bagging prevents particularly, yes, we can say that is what? Undesired cross-pollination. Emasculation prevents undesired self-pollination, because it prevents the self-pollen to get pollinated. And bagging prevents what? We have to add a desired pollen grain. We have to dust a desired pollen grain. So undesired cross-pollination, undesired pollen grains are prevented due to the bagging. Okay, dear students. After artificial hybridization, we will be dealing with the another topics, and in another topic we will be talking about the post-fertilization structure and events. And in this post-fertilization structure and event, if we talk, we will be dealing with the two important events, that is endosperm, about the two important events, that is endosperm and embryo. Okay, so I hope you all are thorough with this. You all understood till now. Now we will be starting with the post-fertilization structure and events. So let's start. Post-fertilization structure and events. In this, we will be talking about the development of endosperm and embryo. Then we will be talking about the maturation of ovule into the seeds, and then ovary into the fruit. Dear student, we will be talking about these things. You should mark this point. What we are going to talk. First, we will be talking about endosperm development, then embryo development, then maturation of ovule into the seed, means we will be talking in the later part about the seed. Then we will be talking about conversion of ovule into the ovary into the fruit. We will be talking about a fruit, how that is formed in this post fertilization events. So you can see seed are produced. So it is a part of post-fertilization event. Okay. And we will be talking about this conversion as well, that is into the fruits. And first we will be starting with endosperm. So by hearing a term endosperm, you should understand that it is a nutritive tissue. And first and foremost thing that should come in our mind, that why it is formed within the seed. Actually it is formed within the seed to provide a nourishment, to provide a nutrition to the developing embryo. And it is developing actually from the primary endosperm cell or primary endosperm nucleus. So this endosperm are triploid in case of angiosperm. So it is a nourishing tissue, nutritive tissue, dear student. And it is developing from the primary endosperm cell or a central cell of embryo sac, means it is a product of triple fusion. Okay. Now you can see it is generally triploid tissue because it is formed after fertilization, and the cells of this tissue are filled with the reserve food material. So it is a nutritive tissue. You can see in this pictorial diagram the yellow colored part is shown as an endosperm. Why? Because it is a nourishing tissue, it is a developing tissue, it is rich in food. Then for nourishment of what they are actually formed, then that answer is the developing embryo. The answer is developing embryo. For providing nutrition to this endosperm is getting developed. Then it is not such that endosperm is found in all angiosperm. It is absent in some of the families like Orchidaceae. It is a family of orchid. Then another one is the Podostemaceae. You can see here this endosperm is absent in which families? That is Orchidaceae, Podostemaceae and Trapezaceae. Why there is no need of this endosperm in these families? Because they have a reserve food stored in their cotyledons, and if there is already reserve food is present in the embryonal cotyledons, then there is no need of this particular endosperm. So it is absent from the start. You can see the particular diagram of this plants of these three families. Next, now if we come to know that this endosperm is present, it has a reserve food, then this reserve food is made up of what? The question should arise in your mind. And if we go on talking about this endosperm, then we come to know that in cereals like rice and wheat, maize, castor and coconut, ivory palm and date palm, the content of this reserve food endosperm is different. So what is the nature of endosperm in this all particular plant? Then you can see that in cereals, that is wheat and rice, you will come to know that in cereal and rice, the particular food endosperm is starchy. Whereas in cereals like maize, which is a monocot, rice and wheat are also one monocot, but in this maize you will find that the particular endosperm nature is proteinaceous. Same way in castor and coconut, we will study about coconut as well. In coconut there is a particularly oily endosperm is found. Okay. Then in ivory palm the cellulosic endosperm is found, and in date palm hemicellulosic endosperm is found. So dear student, here you should note down this is the important question from this can be asked, like match the following. and they can ask that what is the nature of endosperm in coconut, then you should be able to answer that it is an oily endosperm. Okay, sometimes you can get a direct straightforward question, or you can also get a match the following question. Okay, I hope you all are getting this properly, and you should remember this important thing of maize, that a aileron layer in maize is proteinaceous. It is made up of protein. Okay, so let's talk further. Now, it is also known that in some of the particular plants, the endosperm is stony. Endosperm is stony. Means it is a hard, it is a tough. So it is found that this stony endosperm is present in betel nut, that is known as areca nut, and in date palm also it is found to be stony, that is Phoenix dactylifera. Okay, so it is a stony endosperm, this found in case of these two particular sets. Then, and that is obviously in case of seed. Now usually we have studied that endosperm is a triploid in case of angiosperm because it is formed after triple fusion. Whereas in case of gymnosperm we have studied that it is haploid because it formed before fertilization. But in case of angiosperm itself, it is also found that diploid endosperm is found in case of Unothera. That is one of the important example, dear student. It is one of the important example. You should remember that in Unothera the endosperm is diploid. Direct fact based question you can get from this particular part. Okay. Now dear student, this is some initial general information about the endosperm, but now we'll be talking about the type of endosperm. And this endosperm, if we see, are of different type. We have studied that this endosperm represent female gametophyte in case of gymnosperm, but in angiosperm it is a special tissue which is formed after the triple fusion. In angiosperm, based upon the mode of its formation, it is of three types. So we are going to talk about here three types of endosperm. And this three type of endosperm are formed based on its mode of formation. And based on its mode of formation, they are of three types, in that is nuclear endosperm, cellular endosperm, and halobial endosperm. In this, we are going to talk about the first type, that is nuclear endosperm. Dear student, in case of nuclear endosperm, what happens? Karyokinesis is not immediately followed by the cytokinesis. Okay, what happens? Karyokinesis is not immediately followed by the cytokinesis. So you can write karyokinesis means nuclear division is not immediately followed by cytokinesis. So what happens here? Suppose this is a primary endosperm cell, a central cell, and this is primary endosperm nucleus. Actually it is primary endosperm cell, and this is primary endosperm nucleus. Okay. Now if we go on talking about this, okay, so first we correct it. This is primary endosperm nucleus, and this is primary endosperm cell. Okay, now in this karyokinesis will occur, so there will be a 2 nucleus, then there will be a 4 nucleus, and there will be a later on multiple nucleus will be formed within this single primary endosperm cell. Okay, so here a nuclear division occurring repeatedly without the wall formation, so large number of free nuclei are observed, but at the end you will find initially it does not undergo cytoplasm division, but at end cytokinesis will occur, and we will see the formation of several cells within this particular tissue. So this multipolar tissue will be formed, and that endosperm is nothing but a nuclear endosperm. Okay, now I hope you know that in coconut, in central region you will find a water of coconut. So that water of coconut which we drink, that is an example of this endosperm. So watery or milky liquid endosperm is example of this nuclear endosperm. So next we talk about example, that is water or milk of coconut. Okay, and this milk of coconut is of particularly multinucleated condition, so it is a liquid in state, so it is also known as liquid syncytium. It is also known as liquid syncytium. Okay, and this nuclear endosperm is most common type of endosperm. Now apart from that, the next type of endosperm is a cellular endosperm. So next we are going to talk about cellular endosperm. I hope you understood nuclear endosperm properly. Here the nuclear division is not immediately followed by cytokinesis. But when we talk about cellular endosperm, you will know that here the karyokinesis, that is nucleus division, is followed by cytokinesis. And if karyokinesis, nuclear division, is immediately followed by cytokinesis, so the endosperm will become cellular from the very beginning. So what when I put? Suppose this is a particularly primary endosperm nucleus and this is a primary endosperm cell. In this, the nuclear division will occur and two nucleus will be formed. But with that, the cytoplasmic division will also occur. Now this two nucleus of this particular cell is going to form a 4 nucleus. But in that also cytokinesis will occur immediately. So at the end, you will find from the very beginning, the cytokinesis has started already. So because of this, the particular condition like this is observed in case of this type of endosperm. So it is cellular endosperm. This is the second most common type of endosperm after nuclear endosperm. And if we talk about examples, Datura and Petunia are the examples. Are the example of this particular type of coconut, this particular type of endosperm. And this cellular endosperm is known to be mostly confined to dicot families. Okay, so you should remember this. That is the important statement. Now after this cellular endosperm, the third type of endosperm which we are going to study, that is halobial endosperm. Let's talk about the third type of endosperm, that is halobial endosperm. Now in halobial endosperm, one interesting thing is that you will find some mixed things of nuclear and cellular endosperm. Suppose this is a primary endosperm nucleus and this is primary endosperm cell. First what happens, the mitosis occurs and this mitosis is unequal and in this unequal mitosis, complete mitosis is going to happen, that the nuclear division will immediately followed by the cytoplasm division. So two cells like this will be obtained. One is large cell, one is small cell. We can call this a micropylar cell and this is chalajal cell. Now after this, the multiple karyokinesis will happen and multiple karyokinesis happens, so multiple nucleus will be found in these both cells. Means after this cellular type of division, from here it is going to form a nuclear type of division, nuclear endosperm type of division in this both cells. And then at the end, cytokinesis will happen and this type of endosperm will be formed. Then this is known as alluvial endosperm. Okay, so first perfect cellular division, like a cellular type of endosperm division. Then the division of karyokinesis but not cytokinesis, means it follows the nuclear type of division, nuclear endosperm type of division. And at the end the wall formation will happen. Okay, so this type of endosperm is restricted largely to the monocots. Okay, you can write an example, one that is known for this, that is S4-Dallas, and mostly you remember that mostly in monocot this type of endosperm is going to be found. Okay, dear students, so these are the three types of endosperm, that is the nuclear, then cellular, and then alluvial, based on their pattern of development or formation. And I hope you understood now. From this part we will be talking about another two type of endosperm in the mature seeds. Till now we have discussed based on the form of development. Now these are the type of endosperm which are present in mature seeds. Some seeds, mature seeds, are of two types: non-endospermic, which are also known as X-albuminous. We remember now non-endospermic and X-albuminous. Albuminous means the term comes from the protein, that is albumin. Now non-endospermic seed are those in which developing embryo have consumed all the endosperm. Means endosperm is completely consumed by the developing embryo, so embryo is not present. So this type of seeds are known as non-endospermic or X-albuminous seeds. So endosperm is not there. Means this non-endospermic seed are mainly pea, bean, groundnut. Okay, so here the examples are given. You should remember gram, pea, groundnut. Okay, we can write some more example like bean. We can also write some more example like sunflower. These are non-endospermic or X-albuminous. Now why endosperm is completely absorbed by growing embryo? Because the requirement of energy is more by the developing embryo. Now the remaining food somewhat, if endosperm is not there, then remaining food gets stored in the cotyledons. While the another type of seed are endospermic seed or albuminous seed. Again the term albuminous from the protein, that is albumin. Now this endospermic seed have an embryo because it is not consumed completely by the developing embryo. So the endosperm remained persistent in the seed. So this seed is known as endospermic or albuminous seed. And it is found in a case of monocot, dicot, okay, and monocot, castor is also there. In bean it is written here, but bean, some particular different type of beans are there. Some special beans are there in which the endosperm is found to be present. Usually they are non-endospermic. Apart from this, in this seed the food is stored in endosperm. Now you can see the example of endospermic seed like wheat, maize, barley, sunflower and coconut. Sorry, I written sunflower by mistake in non-endospermic seed. You remember this: wheat, maize, barley, sunflower and coconut are the example of endospermic seed. You can see wheat, maize, then sunflower, barley, coconut. Okay, so these are the particular one. Now next, after the endosperm development and type of endosperm, next we are going to talk about the embryo. Dear students, in embryo we are going to talk about the embryo development from the zygote. So the development embryo from zygote is known as embryogeny or embryo development. In this, you know that zygote is formed towards the micropyla end, because an embryo sac egg is situated toward the micropyla end, so obvious zygote gets situated over the micropylar end of some embryo sac, and obviously embryo get developed from that, so embryo should also be present at the micropylar end. So embryo develop at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after a certain amount of endosperm is formed, and zygote will give rise to the embryo. And after, see, first endosperm will develop, then embryo will develop. Why? Because when endosperm get developed, it has a nourishing nature, so the embryo will get it and that will start to divide. So it is an adaptation to provide a proper nourishment to the developing embryo. So here, this is the important statement. You should focus here, and sometimes you will get a question also from this. The endosperm development precedes the embryo development, means it occur before the embryo development. Okay, so this is the most important statement with respect to the question. You should remember that. Now, early stages of embryogeny are similar in both monocot and dicot, and these are given in beautifully in this diagram. See, here you can see properly in this diagram what is happening. This is actually dicot embryo development. See, initially what, there is a zygote, only single cell is there which is diploid. Zygote is undergoing into the two types in the asymmetric division, and two cells are produced. One is terminal cell and one is basal cell. This one is terminal cell, one is basal cell. Now this basal cell is going to form a suspensor, but this terminal cell is known as embryonal cell, and this terminal cell, which is known as embryonal cell, that is going to form an embryo. Now this So here, this embryonal cell is going to form an embryo. Now see, this terminal cell undergo division and it will form a two-cell embryo. Okay, but this basal cell is going to undergo only transverse division. It is going to form what? Transverse division. And in transverse division only it will go and it will form a suspensor. Whereas this two-cell embryo will undergo division and it will form 4, then 4 to 6, then 4 to 8 cell will be formed from here, and this C 2 to 4 and 4 to 8, 8 to 16 and 16 to then a multiple number of cells will be found in this form, and this is given a globular shape. So it is known as a globular embryo. See, this embryo is developed from which one? Terminal cell. But what happened in case of basal cell? Basal cell undergone just transverse division, so it formed a suspensor. And in the suspensor, the last cell is the particular one, a larger one. Then after globular embryo, it will particularly develop this type of structure, a heart-shaped depression you will be observed. So heart-shaped embryo will be formed. Now see, the first cell of this particular suspensor is known as hypophysis. See, this cell is known as vesicular cell, but this cell is known as hypophysis, first cell. Then the particularly heart shape will convert into the torpedo stage, and this torpedo stage will form a particularly mature embryo. Okay, so this is the sequence, okay, in the embryo development in case of dicot. We will discuss, this is just initial info for you. Now how this happens, we will discuss in now. Okay, see, so what happened, just I told you that what was there was just a zygote, and zygote undergo transverse division and two cells are produced. And in that, just we have seen that this is known as terminal or now this is known as terminal or embryonal cell, and this is formed towards the chalaza, whereas this is a basal cell or suspensor cell. We call it a suspensor cell, and this is towards micropylate. Now what happens to this terminal end first? It undergo particular division, and that is vertical division. So if it is undergo vertical division, the two cell will be produced. Then again it undergo vertical division. So this two division, but this vertical division is at a right angle to first one. Now if it is at right angle to first one, you can see I am drawing here a particular cells in a square mode. This was a two cell, but if the another vertical division will occur here like this, then you can see this is the one cell, this is two cell, this is third cell, and this is fourth cell. Actually these are the one cell, and this is the two cell. At the end side you will find this two cells. So this is a what? This is a two celled stage. And this is a four celled stage. Now after this four celled stage, this undergo division, that is a transverse division. And when transverse division will occur, you will see this way. Like this. So what? These are the upper four cells. Yes or no? And this upper four cells are there. Sidewise also you can see this nucleus. Okay. So this two cells, and same way this two cells completely are there. So four cells. Below also this two cells are here, and two cells are here. One behind this one. So total, obviously you will see that total 4 cells on the upper side and 4 cells on the lower side. So total 8 cells stage will be observed. And after 8 cells division, periclinal division will occur. Now this periclinal division is parallel to periphery. So see what will happen like this. The diagram will occur. I hope you understood now how this 8 cell condition as above observed. But if the division which occur parallel to periphery like this, so what will happen? Here one cell, here one cell, one cell here, one cell here. So total 8 cell this side, 8 cell back side. Yes or no? So here you can see this upper octet is there. This is the upper octet. And in upper octet you will see that particularly 1, 2, 3, 4 cells are there, and 1, 2, 3, 4 cells are there. So 8 cells are there. That's so it is upper octet. And this is lower octet. So 8 or 8 16 cell stage is there. And in this 16 cell stage, this is a upper octet. And this is a lower octet. Okay, like this up to 16 cell division will occur, and then in this several mitosis will occur, and you will see the formation of the globular embryo. So many cells are formed in this. So we see the globular embryo after this many stages. And this suspensor cell undergo transverse division, several transverse division, and several transverse division will occur simultaneously with this at the bottom side, and at the end in the globular embryo you will see so many cells are found to be attached with this. The last one is the larger cell, and this is known as suspensor. In this suspensor, the first cell which is attached to the globular embryo is known as hypophysis, and this cells, the bottommost cell which is exactly at the micropyle side to the globular embryo, that is known as vesicular cell. Okay, so this way the particularly suspensor will be formed. Now according to NCERT, you should remember that this suspensor is 6 to 10 celled. How many cells are there? 6 to 10 cells embryo. 10 cells are found in case of the embryo. Okay, so this way the particularly whole, okay, the embryo will be formed. And you should remember that suspensor cell undergo transversor and form suspensor, and you can see suspensor is located here. After this 6 to 10 cell suspensor and globular embryo, several division will occur and the further stages will be observed. And in further stages you will see that there is a heart shaped stage. Okay, so we will say like this. So many cells are there. And here you will see 6 to 10 celled suspensor. And this is a heart shaped embryo. So it is heart shaped stage. After heart shaped stage, the depression will grow inside, and after this you will find this stage like this. And this is known as torpedo stage. After torpedo, this two extension will elongate, and when this two will elongate, they are going to form mature embryo like this. In this mature embryo, this 2 will be representing what? Cotyledon, and this region will be known as plumule. Okay, and apart from this, you will find that there is a presence of here a particular root cap part. You will find a radical, and here you will find hypocotyle region, and this is the structure of mature embryo which is given in NCERT. You should remember this labeling according to NCERT. It is very much important. Okay, and suspensor get dissolved. So this is the dicot embryo, mature embryo. Now if I want to make some of the understandable diagram, then if someone asks draw one of the understandable diagram of this particular, which is showing a labeling of all the particular parts, then we will draw such diagram, such simple diagram which is representing all the parts. Okay, so this is known as plumule. This is the epicotyle. These are cotyledons. This is hypocotyle, and this is radical. So you know that the tapering at below is the radical, the tapering at the above is the plumule, and this hole from here to here, it is known as embryonal axis, and this embryonal axis is also known as tigellum. And the epicotyle, which is given here, that can be defined as it is a region of embryonal axis between the plumule and the cotyledon attachment site. And hypocotyle can also be defined as it is a region of embryonal axis between the cotyledon and the particularly radical. So it is one of the schematic diagram of dicot embryo to represent the different parts of this particular one. One more thing you should remember, that if it is a schematic diagram of typical dicot embryo, then this in future this plumule will form a shoot. It will form a shoot, and this radical will form a root. Okay, so in future this will happen. Okay, I hope you understood the development of embryo perfectly. So stages of an embryo development in dicot. Okay, so first you note down. I am removing this, then we will continue further. Okay, so let's first start for the stages in embryo development. We have discussed this in detail. Now I hope you are able to understand these all stages, because we have shown in a typical diagram as we study in a regular classroom. This is a dicot. A typical dicot embryo consists of embryonal axis, tigellum to cotyledon. We have discussed this. The portion of embryonal axis above the level of cotyledon is the epicotyle, and the terminates with the femule or a stem tip, that is a first apical bud of shoot. The cylindrical portion below the level of cotyledon is hypocotyle. Just we have discussed that. The terminate to its lower end, it is known as radical or root tip. The root tip is covered with the root cap. You know that. Okay. The typical dicot embryo diagram is given here. It is NCERT diagram, so you should remember this. The particular parts, because in so many times they will ask a question of labeling from this. Okay. So this is the plumule. Okay. You can see these are the two cotyledons. Okay. This is one. This is two. Then this is hypocotyle. This is a radical as we have shown. This is a root tip. And this is the most important question with respect to labeling you can get in question exam. Now apart from that, next we are going to talk about monocot embryo. Embryos of monocot possess only one cotyledon, and the monocot are the grasses. So in grasses family, the single cotyledon which is found in monocot embryo, it is known as scutellum. And one of the peculiar feature of this scutellum is this, that this scutellum is situated towards the one side, that is lateral side of the embryonal axis. Where it is located? On the lateral side of embryonal axis. We will discuss this in with a diagram. At the lower end, the embryonal axis is the radicle and the root cap, and it is enclosed in the undifferentiated sheath called a coleorhiza. We will discuss where the coleorhiza is there. The portion of embryonal axis above the level of attachment of scutellum is epicotyle, and epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, that is a coleoptile. First, let's talk about diagram, then we will discuss this particular one, then you will understand it properly. This is a particular diagram of embryo of grass or monocot, and you should focus that in this embryo you can see this is a scutellum, that is a single cotyledon. This is coleoptile, okay, which are the protective covering of a shoot apex. And here you can see at the bottom side you can see this is a radicle, and this radicle is covered by the root cap, and here coleorhiza is a protective covering of the root cap or radicle. Here you can see, you have seen this is a scutellum, that is a single cotyledon, and this is epiblast. Now epiblast represent remains of second cotyledon, because it is known that initially dicot have evolved and dicot had a two cotyledon, but when evolution occurred and monocot evolved, monocot had a single cotyledon, or they have now a single cotyledon. So evolution occurred from two to one cotyledon, so one cotyledon has lost, so the remains of that must be present in the particular embryo of monocot, and thus remains are showed of a second cotyledon is shown in case of epiblast. Okay, so you should remember this. Now, so before understanding this, we should read again this statement, so the monocot embryo will be defined perfectly. Dear student, as we have seen in the diagram of this monocot embryo, you understood that it possesses one cotyledon, that is scutellum, on lateral side of embryonal axis. At lower end in diagram, we have just seen that embryonal axis are the radicle and root cap, and that is enclosed in an undifferentiated sheath called coleorhiza. We have also seen portion of embryonal axis above the level of attachment of scutellum, and we called it a epicotyle. You can go back and study this diagram again. Epicotyle has a root apex and few root leaf primordia, initial leaves enclosed in a hollow foliar region, that is a coleoptile. Then outer covering of endosperm separates the embryo by a proteinaceous layer, that is a linol layer. We remember when we see this embryo in a seed, of the particular embryo, you will see that the endosperm is separated from the embryo by one layer between them, that is a proteinaceous layer, that is called as the alinor layer. And endosperm is obviously, you know, there's 3N, so here alinor layer is also 3N and proteinaceous. You should remember this. We will discuss this part when we will study seed in the next class. Okay, so diagram we have seen this but now perfectly, so you remember now epiblast as well. Then the one more thing which we are going to discuss in this continuation of this lecture, that is perisperm. Now what happens when embryo development occurs or endosperm formation occurs? Mostly nucellus get consumed after fertilization due to the absorption of food by endosperm and embryo. But there are some seeds in which you will still see the presence of nucellus after fertilization. Then this type of seed with the persistent nucellus are known as perispermic seed, and that is known as perisperm. So perisperm is found in some seeds, and some seed remnant of nucellus are found that are persistent in nature, and this residual persistent nucellus is known as perisperm. So you remember this takes two terms. What is perisperm? Perisperm is a persistent nucellus after fertilization in a seed. Then you can see this is a perisperm, radical break and embryo is there. You can see the presence of perisperm. Now such seed are known as perispermic seed, and the example of perispermic seed are black pepper and beet. Black pepper is also known as Piper nigrum. You can see the diagram of this, that is a black pepper is given here. Then another one is the beet. Beet also has a perisperm, that is persistent nucellus. Okay student, so I hope you are now clear with the particular topics like endosperm, embryo development, types of endosperm, all this we have discussed. So we have still studied development of embryo and development of endosperm, not of the structure of seed, which we will study in the next class. Before that we should discuss some of the questions. And in first question which we are going to study, that is the cells of endosperm are filled with the reserve food material and are used for the nutrition of developing embryo is generally blank in angiosperm. So you know that they are the triploid tissue developed by the triple fusion, and in triple fusion the fusion of secondary nucleus and male gamete occurs. So it is a triploid tissue. Then next question is the portion of embryonic axis above the level of cotyledon. You know that just we have studied above the little level of cotyledon you will find the presence of epicotyle. Okay, and below the cotyledon you will find a hypocotyle. Next to that, primary endosperm nucleus, that is pen, develops into, you know that it develops into endosperm. Then perisperm is found in the seeds of, you know that perisperm is nothing but persistent nucellus, just we have studied. It is found in Piper nigrum, we have just studied black pepper. Next question is in gram, pea and groundnut plants, endosperm is not found. You know that gram, pea and groundnut are non-endospermic seed, and if they are non-endospermic seed, then if we go with the statement, first statement is given is endosperm is not formed in this life cycle. Yes, endosperm is not formed. So if we see, the endosperm is formed, but in mature seeds endosperm will be absent, so they are non-endospermic seed. Mature seeds lack endosperm. Remnant of nucellus in the seed is seen in the seed is, you know, the remnant of nucellus or persistent nucellus is known as perisperm. Now I hope you are clear with that. Okay dear students, so till now we have discussed in today's lecture about the endosperm, embryo, artificial hybridization, and I hope you are now clear with this. Thank you, thanks a lot. We will meet in next lecture with the different type of seeds. Okay, till that, have a good day.